Guys, we're not loud enough. We have to go even louder, all right? Ready, set, go.
Santa. 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 Santa.
Guys, one more time, even louder.
It's okay. Go right. Go get your scooter. Robert, go get the scooter, please, for Bella. On Monday, October 18th, 2021, at 8:45 a.m., we got on a Zoom call with genetic counselor Rachel Palmquist from Primary Children's Hospital. The call was scheduled to learn about our recent Exome Clinical Sequence test done on Isabella, Courtney, and myself, to see if she had a genetic mutation, a deletion, or something else. Rachel shared terrible, heart-wrenching news. Isabella is 1 in 20 to have a rare genetic deletion of her Trip12 gene. This news hit us like a ton of bricks. We didn't know what to do. Courtney and I were not equipped to hear this news or to fully understand what it means. As I started researching what this meant for Isabella, my anguish for her felt like nothing I've ever experienced in my life. It felt like the floor just fell out from underneath me. I was in a free fall for the next few days. Isabella had a rough start to this life. On October 16, 2016, she was delivered via emergency C-section because her umbilical cord was wrapped around her neck. During her time in the NICU, she didn't have any visible signs of brain damage, but she struggled to feed and to breathe without oxygen support. After 3 weeks in the NICU, Isabella was cleared to go home, and she had a clean bill of health. We had no reason to believe she had any genetic or underlying health conditions outside of being a normal preemie baby. We were met with some health challenges shortly after her first year. Isabella suffered from global developmental delay, strabismus, which is an abnormal alignment of her eyes, trichotillomania, which is excessive holing of her hair, tremors, and ongoing ear infections that lasted years on end. Tubes helped very little, and she would get at least one ear infection each month, and that would last for about 2 weeks, and this went on for years. We attempted to take on each of these conditions individually, but it was the daily tremors that concerned us the most, aside from her developmental delays, which we thought she would quickly grow out of. We visited a neurologist to discuss her tremors and went through a variety of different tests that were all inconclusive. As a last-ditch effort to find answers, we dropped $5,000 to get a genetic test, and what do you know? Nothing. It's not genetic. At least that's what they thought and what we thought. During this same time in her life, we were visiting Primary Children's Hospital to find out why she was having severe speech delays and the lack of cognitive growth. Test after test, and tens of thousands of dollars later, to only have revealed no new insights. No answers. All the while, Isabella's health is worsening, and new conditions keep adding to the stack. Once COVID-19 hit in early 2020, most tests and office visits became a thing of the past, and our journey to find answers was halted. As parents, we felt defeated and hopeless of ever finding answers. So for now, we were just focusing on treating the symptoms. Then, in the fall of this year, we decided to act on the recommendation of getting a full exome clinical sequence of the three of us, a test that we weren't familiar with. We found that this test is different from your normal genome test that we did 2 years earlier. Clinical whole exome testing is a test for identifying disease-causing DNA variants within the 1% of the genome which codes for proteins or flanks the region which codes for proteins. It's not the cheapest test to conduct, $22,000, and not a lot of support from health insurance. After years of seeing a variety of specialists and getting nowhere to finding the root cause of her condition, we decided to try the clinical exome sequence analysis, and that changed everything. You know, a few people have come to me and, you know, they've said, Stephen, aren't you so glad that you know what's wrong with Isabella? Like, you've got the diagnosis. And you know, part of me is grateful that we have a diagnosis because now we can take an educated approach to her health. But unfortunately, her diagnosis, this deletion of the Trip12 gene, there's not a lot of research. There's only one research paper on the topic. It's a fairly new discovery, and there's not a lot of people in the world that have this rare deletion. And so I had to purchase several textbooks on biochemistry, molecular biology, genetics, to really try to understand what's going on with Isabella. And the more I've learned, the more troublesome it is, the more bad news around every corner. And so I want to explain what I've learned so far about Isabella's Trip12 gene deletion. And I am not a geneticist or an expert, but I want to share with you what I've learned so far, and I'll continue to share as I learn. So let's start from the beginning. Let's start with DNA and go from there. All right, so what I'm about to show you is an accurate representation of the actual DNA replication machine that's occurring right now inside of your body. So the DNA is entering the production line on the left here, and then it hits this collection. These are miniature biochemical machines that are pulling apart the DNA strand and making an exact copy. So DNA's in and it hits the blue donut-shaped structure, and then it's ripped apart into two strands. One strand can be copied directly. As you can see, these things are spooling off to the bottom there. But things aren't so simple for the other strand because it must be copied backwards. So it's thrown out repeatedly in these loops and copied one section at a time, creating two new DNA molecules. Now you have billions of these machines right now working away inside of you, copying your DNA with exquisite fidelity. So again, here's your DNA. It's just jiggling and wiggling here because of the surrounding soup of molecules, which has been stripped away so you can see something. But DNA is about 2 nanometers across, which is really quite tiny. But in each one of your cells, each strand of DNA is about 30 to 40 million nanometers long. So to keep the DNA organized and regulated access to the genetic code, it's wrapped around these purple proteins. It's packaged up and bundled up. All this field of view is a single strand of DNA. This huge package of DNA is called a chromosome. And we'll come back to a chromosome in a second here, but we're pulling out, zooming out here, and through a nuclear pore, which is the gateway to this compartment that holds all the DNA, called the nucleus. This is the way a living cell looks down a light of a microscope, and it's being filmed under time lapse so you can see it moving. The nuclear envelope breaks down. These sausage-shaped things are the chromosomes. They go through this very striking motion that is focused on these little red spots. When the cell feels it's ready to go, it rips apart the chromosome. One set of DNA goes to one side and the other set goes to the other side of the DNA. Identical copies of the DNA. And then the cell splits down the middle And again, you have billions of cells undergoing this process right now inside of you. Now we're going to rewind just a little bit and focus on the chromosome. And look at its structure. So again, we're looking at this equator moment. The chromosomes line up. And if we isolate just one chromosome, we're going to pull out and take a look at its structure. So this is one of the biggest molecule structures that you have, at least as far as what people have discovered so far inside of us. This is a single chromosome, and you have 2 strands of DNA in each chromosome. One is bundled up into one sausage, the other strand is bundled up into the other sausage. These things that look like whiskers are the dynamic scaffolding of the cell. They're called microtubules. We're going to focus on the region labeled red here, and it's the interface between the dynamic scaffolding and the chromosomes. It is central to the movement of the chromosomes. We have no idea as to how it's achieving this movement. People have been studying this thing called the kinetochore for over 100 years with intense study. It is made up of about 200 different types of proteins, thousands of proteins in total. It is a signal broadcasting system. It broadcasts through chemical signals, telling the rest of the cell when it's ready. And when it's ready, it turns green like this. Really cool. And you see the separation of the chromosomes. I mean, look at this. It's beautiful. You have these that are walking back. I mean, it's mechanical. It's molecular clockwork. This is how you work at the molecular scale. So it's pretty neat. These carrier molecules are walking away. On this note, let's understand chromosomal mutations. As the name says, chromosomal mutation means the mutation of chromosomes. And how exactly does it get mutated? Well, it can happen in a couple ways. One way is where the structure of the chromosome gets disrupted. The other is where the number of chromosomes are different than the usual in the organism. Let's take a look at each type. This is the structure of a typical chromosome. Now imagine this small parts gets physically removed. This would result in a chromosome in getting smaller as the segment is lost. This also means that if the segment had some important genes, that they will also get lost, which can result in severe abnormalities. Such type of mutation is called a deletion. This would be true for Isabella's case. Now imagine this part of the chromosome gets duplicated. As a result, the chromosome's length increases. Needless to say, this will also result in some of the other abnormalities in the individual. This type of mutation is called duplication. Now comes the next type. These are 2 different gene segments. Imagine they get flipped. Thus, the chromosome will appear like this. This is called inversion. It may or may not be harmful, depending on which gene gets flipped. Now let's take one more chromosome. Imagine a part of this chromosome gets removed and gets inserted in this chromosome. Both the chromosomes will change drastically. This type of mutation is called an insertion. And now comes the last type. Instead of a segment getting inserted to another chromosome, let's imagine a situation where a segment from each chromosome gets interexchanged. So this segment will get into this chromosome and vice versa. This kind of mutation also results in abnormalities as both chromosomes get altered completely. This type of mutation is called translocation. All these mutations, resulting in the disruption of normal chromosomal structures, are broadly capped under a single heading. They are categorized as chromosomal aberrations. Hopefully you've learned some basics of DNA and how it's replicated. I know I sure have. It's been very eye-opening. But it's important to understand the foundation of DNA as we move into this next section to talk about the Trip12 gene and the deletions of Isabella's exons 37 through 41. It's a lot to comprehend, and even more challenging is the expression of these exons 37 through 41, the phenotypes, what that means for Isabella and her health. So let's jump right into it. Mutations or deletions in the Trip12 gene, and the long name for the Trip12 gene is called the thyroid hormone receptor interactor 12, which encodes the E3 ligase in the ubiquitin pathway. This has been associated with the autism spectrum disorder. In addition to autistic features, the Trip12 mutation carriers show moderate to severe intellectual disability. More recently, Trip12 was postulated as a novel candidate gene for intellectual disability in a meta-analysis of published ID cohorts. It is not surprising that many members of the ubiquitin pathway have been associated with diseases including cancer such as BRCA1, the associated breast and ovarian cancer, neurodegenerative diseases such as Parkinson and Alzheimer's disease, neurodevelopmental phenotypes in autism spectrum disorder. Hence, mutations in members of the ubiquitin pathway, and especially E3 ubiquitin ligases, are important contributors to the neurodevelopment pathophysiology. The identification of novel genetic causes of intellectual disabilities and autism spectrum disorder have been the subject of intense research. First thing, we will jump into something called the ubiquitin pathway. The ubiquitin pathway is an enzymatic cascade including activating E1, conjugating E2, and ligating E3 enzymes, which govern protein degradation and sorting. It is crucial for many psychological processes. Compromised function of members of this ubiquitin pathway leads to a wide range of human diseases, such as cancer, neurodegenerative diseases, and neurodevelopmental disorders. For cells to work properly, there must be a healthy balance of unwanted proteins being broken down and new proteins being made from them. This recycling process is necessary to prevent damage or surplus proteins from building up and becoming toxic to the cell. It relies mainly on structures known as proteasomes. Any unwanted proteins attacked with a chain of proteins called ubiquitin. Any protein with a ubiquitin tail means that it's destined for a proteasome. Proteasomes are shaped like barrels. They open at one end to allow tagged proteins to enter, and at the other end allow broken down pieces of the protein to exit only. Tagged proteins are captured by proteasomes. Once captured, the protein unwinds and enters the proteasome inside. The broken down pieces are then released and recycled to make new proteins. Sometimes proteasomes can be overwhelmed when cells generate large amounts of unwanted proteins. When this happens, a different clearing system is used. Instead, tagged proteins aggregate into structures called egg reserves. These are cleared by other structures called lysosomes, which get rid of the proteins instead of recycling them. As I've learned more about the Trip12 gene and Isabella's missing exons 37 through 41, her partial deletion, it's been pretty overwhelming. From the surface, individuals with Trip12 deletions or mutations can have developmental delay, extremely short stature, small hands, dysmorphic facial features, hearing loss, and epilepsy. But there's a lot more at stake due to the transcription factors and eventually what could be the lack of cell degradation. The predicted 1992 amino acid Trip12 protein contains a putative transmembrane domain. Analysis detected Trip12 expression in all 16 human tissues, with the highest expression in skeletal muscle and testes, lower expression in the heart, kidney, spleen, thymus, prostate, ovaries, placenta, and white blood cells, and other low expressions in the brain, lung, liver, pancreas, small intestine, and colon. The thyroid hormone receptors are hormone-dependent transcription factors that regulate expression of a variety of special target genes. They must specifically interact with a number of proteins as they progress from their initial translation and nuclear translocation to heterodimerization with retinoid X receptors, functional interactions with other transcription factors, and the basic transcriptional apparatus, and eventually degradation. It's interesting to learn the protein expression of this gene of Isabella's exons 37 through 41. They interact with the following with a median read count per base of 3.2 to 9.9, testes, cells, cultured fibroblasts, liver, kidney, whole blood, spleen, pancreas, brain, frontal cortex, kidney cortex, heart, left ventricle, brain spinal cord, brain cortex, brain cerebellum, brain cerebellar hemisphere. So in summary, the Trip12 gene is part of the HECT domain E3 ubiquitin family, and the Trip12 protein is expressed in multiple tissues and thought to play an important role in a number of processes, including ubiquitin fusion degradation and regulating DNA repair and cell growth. So as a result of this gene deletion and others that have the gene mutation of the Trip12 gene, there's definitely a strong presence of neurodevelopmental issues. There's developmental delays, speech delays. Some children are nonverbal. Many have autistic behaviors. Many have, hypertonia, seizures, vision problems, hearing problems, OCD, ADHD. You can imagine that when your cells, you have 2 billion cells per day that are recycled, where they're taking out the trash and or they're being completely killed off. 2 billion per day, and this gene is essential to that function. And we still don't know to the extent of the impact that it's having on Isabella. That's something that we need to do more research, and we need to find answers. So answers are what we're going after. Presently there is no cure for Isabella. There's not a lot of research on the Trip12 gene, and that needs to change. This video is part of that call for resources, for researchers that want to learn more about the Trip12 gene and want to help individuals like Isabella that have a deletion or a mutation find some treatments and treatments that can help her. So today I'm announcing a fundraising campaign for Isabella. There will be 2 major rounds of fundraising. The first is today's round, and that is to cover the financial costs of her immediate needs. The second round is to raise capital to hire researchers who will take on the research and eventually develop a treatment or a drug that can go into clinical trials. Today's fundraising goal is $50,000. It's no small amount, and I pray with your help that we can get there. Isabella has some immediate needs for her diagnosis. Children with this gene deletion often suffer from seizures, dysmorphia of their face, strabismus, and are mostly nonverbal, with neurodevelopmental delays similar to autism, and even more rare cases, some children suffer from strokes and rare cancers. Isabella, she suffers from tremors, and it looks like this is just an early onset of future seizures. This is from her Trip12 gene deletion, causing proteins to stop reading the gene before the end of the gene. It's called an early stop codon. In this rare condition, children are diagnosed with epilepsy. We are working on getting her a service dog that can be trained to handle and alert us of these seizures. In addition to recognizing seizures, the dog will provide emotional support for Isabella. Her current dog, Miko, he's on his last leg of life, and he's been one of the most important tools in helping Isabella with her anxiety. He's so sweet. He's been such a good dog for her. And so looking at service dogs and what we need to help Isabella with, we estimate the cost of a service dog to meet her needs to be upwards around $30,000. Next, we need to purchase a custom mouth guard that will need to be replaced as she grows and gets older. Dysmorphia of the face just has not passed Bella. Her jaw is very small and it's not aligned properly with her airway, and so this causes all sorts of issues with breathing at night, problems with chewing her food, and this will be a decade or longer fight to realign her jaw. And this first mouth guard will cost us $10,000, and there'll be more costs down the road as she grows. And then to support her neurodevelopmental delays and nonverbal cues, we are in the need of a tablet and software that can help Isabella communicate her needs with us. Since she suffers from a low IQ threshold, we will also need a behavioral therapist as well as a speech therapist to work with her one-on-one to help her communicate. Unfortunately, her diagnosis is so new and unknown that there's no state-funded programs that she qualifies for. All of this is just the beginning, but with your help in us reaching our $50,000 fundraising goal today, we believe this is going to give Isabella the jump start she needs in living a better quality life. So I hope you'll join us in this event and donating whatever you can, even if it's just $1. That's $1 closer to helping Isabella live a better quality of life. And if you can't donate, please share this out on social media, and maybe just someone that's watching this event because of you is willing to donate, even just $1. So again, please join us in this event, share it out, and let's help Isabella with her quality of life. Let's help her get the things that she needs to live a better life.
Oh, careful. You gotta pedal.
You gotta put one foot on and start pedaling. There you
go. Yeah. I'm so impressed.
So as we continue this fundraising event, I want to share a quick video of an example of how we would like to use this tablet for Isabella. I did mention that, due to this being so new, there's not a lot of support out there, including state funding. Luckily, she's in special ed in the public education system, so she does get some help there during the day. But she needs so much more. She needs after-hours private speech therapy and behavioral therapy, and her therapists have recommended that she use one of these tablets to communicate, and this is really going to help her develop her speech. There's a certain threshold that she may hit with her speech and never exceed that. But we certainly want to push the limits, and this tablet is what is going to help us accomplish that. So please enjoy this next couple minute segment of what we're trying to accomplish with Isabella.
My name is Orion.
Orion is nonverbal, but he has lots to say.
Do it again.
I am 5 years old. I have a brother named Saren.
Using an electronic program called Language Acquisition through Motor Planning, he is able to express himself by selecting pictures, and then the iPad generates his speech.
Drink. Apple juice.
Yes, that's apple juice. Good job. High five.
For Orion's mother, hearing her son's own words for the first time was an emotional experience.
He asked for a banana. Oh, I cried. I cried a lot.
So much so that she now has a tattoo of a banana on her arm.
Everyone waits for their child to talk for the first time. That's one of those big milestones. And this was just an amazing thing that we could do with the tablet.
Before the kids reach the stage that Orion is at with this tablet, they first learn how to communicate with simple images. They're using a system called PEX, or Picture Exchange Communication System.
She told me, I want to pee. Let's go to the bathroom. Let's go.
Once the world of communication is opened up, it allows them to focus on what they want and be understood, and decreases all of those other things.
The children learn to use pictures to indicate what they want, and now that's been extended to learning how to do that digitally.
So where that might be viewed as a bit different 15, 20 years ago, now we're very excited to see kids being very socially accepted, using devices that everybody uses.
And once they get the hang of it, it's amazing how much these nonverbal kids can actually have to say.
What did he just tell you?
That he likes vanilla butterscotch and strawberry ice cream.
So they're able to communicate everything they want to say.
Yes.
Well, there are currently more than 200 children using this technology here at Grandview. The hope for the future is that they'll have their own communication technology lab so that they can offer this to even more kids. In Oshawa, Audra Brown, City News.
So as you can see, this is just a great communication tool for Isabella, and we believe this is going to be a turning point in her communication. She's mostly nonverbal. She knows a little over a dozen words and can't use more than one word at a time. And she, has a threshold. So like the color yellow, it is the color for everything. And then she started saying purple, and we just loved that. And she says it so well, but she does not say yellow anymore. So she'll replace words with another word and doesn't retain that vocabulary. And so this tablet is going to be a big help in that. The next thing I want to discuss is the need for a service dog that not only can handle her nonverbal behaviors, can really handle her anxiety. Miko, her current service animal, puts her to bed every night and is just there for her no matter what and for anything. And so she's going to need that once Miko passes. And then even more of a need for the detection of seizures. This is our biggest worry. This is our biggest concern. She's had tremors her whole life, and that's what really started this journey. And now we're at a point where we could wake up one day finding her having a seizure. And so we want to get ahead of this. And so by doing that, we're going to get a service dog that can be trained to detect and manage seizures and to alert us, so we can stop her from getting more brain damage or even worse, having a stroke. And so that's the biggest part of this fundraiser, is the need for a service dog. And as we've shopped around, the needs that are specific to Isabella could cost upwards to around $30,000, and we're gonna get it for her. We're gonna fight tooth and nail. We're gonna do whatever it takes. And so every dollar counts. If you're watching this and you can just donate $1, that's $1 less that I have to raise or earn to make that happen. I want to share one last call to action, and that is for the U.S. government, the world governments, the WHO, to take a serious look at CRISPR and consider the ramifications it has for those like Isabella who have this gene deletion and could be cured overnight with the proper CRISPR treatment. For those of you that are not familiar with CRISPR, I'm going to share a quick video explaining the details. But there's a new tool out there, the Cas9 protein, that can be used to fix Isabella, to insert the correct genetic code of the Trip12 gene to fix her overnight. And unfortunately, it's not popular among the world governments because of the idea of designer babies and super soldiers. But the technology's out there and it's not going away. And we need to, as a community, as a world, need to take a close look at this technology and find ways where it can be used to help those with genetic deletions and variants that need, these need to be for cases that are more dire and life-saving. I think that's a good place to start. So I want to share this quick outline of CRISPR, and hopefully this is a technology that can be used for Bella down the road.
Every cell in our body contains a copy of our genome, over 20,000 genes, 3 billion letters of DNA. DNA consists of two strands twisted into a double helix and held together by a simple pairing rule. A pairs with T and G pairs with C. Our genes shape who we are as individuals and as a species. Genes also have profound effects on health, and thanks to advances in DNA sequencing, researchers have identified thousands of genes that affect our risk of disease. To understand how genes work, researchers need ways to control them. Changing genes in living cells is not easy, but recently a new method has been developed that promises to dramatically improve our ability to edit the DNA of any species, including humans. The CRISPR method is based on a natural system used by bacteria to protect themselves from infection by viruses. When the bacterium detects the presence of virus DNA, it produces two types of short RNA, one of which contains a sequence that matches that of the invading virus. These two RNAs form a complex with a protein called Cas9. Cas9 is a nuclease, a type of enzyme that can cut DNA. When the matching sequence, known as a guide RNA, finds its target within the viral genome, the Cas9 cuts the target DNA, disabling the virus. Over the past few years, researchers studying this system realized that it could be engineered to cut not just viral DNA, but any DNA sequence at a precisely chosen location by changing the guide RNA to match the target. And this can be done not just in a test tube, but also within the nucleus of a living cell. Once inside the nucleus, the resulting complex will lock onto a short sequence known as the PAM. The Cas9 will unzip the DNA and match it to its target RNA. If the match is complete, the Cas9 will use two tiny molecular scissors to cut the DNA. When this happens, the cell tries to repair the cut. But the repair process is error-prone, leading to mutations that can disable the gene, allowing researchers to understand its function. These mutations are random, but sometimes researchers need to be more precise, for example, by replacing a mutant gene with a healthy copy. This can be done by adding another piece of DNA that carries the desired sequence. Once the CRISPR system has made a cut, this DNA template can pair up with the cut ends, recombining and replacing the original sequence with the new version. All this can be done in cultured cells, including stem cells that can give rise to many different cell types. It can also be done in a fertilized egg, allowing the creation of transgenic animals with targeted mutations. And unlike previous methods, CRISPR can be used to target many genes at once, a big advantage for studying complex human diseases that are caused not by a single mutation, but by many genes acting together. These methods are being improved rapidly and will have many applications in basic research, in drug development, in agriculture, and perhaps eventually for treating human patients with genetic disease.
Thank you, all of you, for tuning in and watching this film and learning a little bit more about Isabella and her Trip12 gene deletion. I hope that this will inspire you to donate. I hope this will inspire you to share this message on social media to bring awareness to the 20,000 plus gene deletions and variants that are out there, and the ones that are being discovered every day, just like this Trip12 gene that was discovered just a few years ago. I sincerely appreciate you watching this, and I'm going to end this short film with just a montage, upbeat montage of Isabella, and with your help, we are going to reach our fundraising goal, this first round of 50,000. And then on the second round, we are going to enlist the help of researchers that will research this gene and really help them develop a treatment or a drug that can help people just like Isabella and many, many more. So enjoy this montage. Thank you for watching, and please share this with your friends and family on social media, and let's bring awareness to Isabella and others like her. I don't know if it'll be live by the time I put this video out, but we are going to have some merchandise where all the proceeds are going to go to her treatments, her current treatments and needs, and beyond. And so if you want to support Isabella and get a Trip12 t-shirt or hat, definitely check out the online store. It may be available in time for this video. If not, look for a future link in the next couple of days. So thank you again. We can't thank you enough, and enjoy this short montage of Isabella. Thank you.
I'm trying to hold my breath, let it stay this way, can't let this moment end. You set off a dream in me, getting louder now. Can you hear it echoing? Take my hand, will you share this with me? 'Cause darling, without you, all the shine of 1,000 spotlights, all the stars we steal from the night sky will never be enough, never be enough. Towers of gold, still too little. These hands could hold the world, but it'll never be enough.